He kōna I purangi tēnei nā te reo irirangi o Aotearoa.
It's been described as kind of a landscape of fear. Everything's trying to eat everything else, and you're trying to eat things but not being eaten yourself.
Kia ora, nau mai haramai ki te ao hurahanga. Hello and welcome to Our Changing World. Ko kalekin kanan tēnei. Professor Jeff Shima is a fish ecologist based at Te Henanawaka, Victoria University of Wellington. And today we're talking about his research focused on a type of fish called lanternfish. He's interested in the complicated dance they perform linked to day and moon light, and how this impacts other fish species in this watery landscape of fear. He's brought some along in a jar so I can have a look.
When you look at them, they're probably smaller than what most people expect. They're kind of similar to what you might expect to see if you're looking at a small anchovy or a sardine. These ones were collected from an island called Tetiaroa in French Polynesia in 2022. The ones that we catch closer to New Zealand, where the water is colder, they tend to be a bit larger. So we get larger-bodied myctophids down here, different species.
The one he has is thumb-sized, about 5 centimetres. In Otago, maybe they get to 10 or 12 centimetres, he says, and they generally don't get much larger than that. So it's not just one fish, it's a family of fishes, Myctophidae, with about 250 species, and they're found throughout the world's oceans wherever there is deep water.
They're called lanternfish because they have these structures on their bodies that produce light. Those are called photophores, and they can use those for a variety of things, including camouflage, communication, possibly for aiding in hunting.
Just to be clear, these are not the guys that live in the deep sea that have the little thingy at the front. Those are anglerfish.
Those are anglerfish. Yeah, common misconception. So the things that have what looks like a lantern hanging in front of their mouth that you probably saw in Finding Nemo is one of the villainous fish. That is a deep sea anglerfish. Also very cool, but not a lanternfish.
And the reason why you and others are particularly interested in lanternfish is because there are a lot of them.
There are, as a group, they're probably the most abundant vertebrate on the planet. They probably make up about 65% of the deep sea fish biomass. And another important aspect of them is they move. They move from deep water up into shallow waters every night, and that potentially has really big effects.
This movement was first discovered through the use of sonar during World War II. Operators were puzzled by what appeared to be a false seafloor about 300 to 500 meters deep during the day and less deep at nighttime.
In regions where sunlight almost ceases to exist, there is an echo of sound. Who or what reflects that sound? This is the story of a search for something in the deep seas, something that creates the illusion of an ocean floor, where no floor can exist. This is the story of an ocean phenomenon known as the deep scattering layer.
The sonar was scattering off the huge numbers of marine critters that migrate up into the shallower water at dusk to feed. It's the world's largest animal migration. It's not just lanternfish in this deep scattering layer, but they make up the majority of it. The worldwide biomass of lanternfish is estimated in the billions of metric tons, which is several times the annual global fisheries catch. Their lives are ruled by light, and they like to hang out in a dimly lit part of the ocean.
It's the depths of the ocean between about 200 meters deep and 1,000 meters deep. And it's the area of kind of perpetual twilight. So when it's light and bright up at the surface of the ocean, the twilight zone is like dawn or dusk, like you can't see very well during the day. And it progresses down to pitch black when you get to 1,000 meters. So that's called the mesopelagic zone. And so that's the habitat that many of these lanternfish live in during the daytime. And so at dusk, when the sun is setting, these animals start to migrate up towards the surface, and they're effectively following smaller animals that are also doing these migrations, things like copepods, small crustaceans that they're feeding upon. And everything is kind of moving up to the surface waters because that's where the highest concentration of food is. And so that's where phytoplankton, the small microalgae, can grow and get access to light. So that's where it's very productive. So they want to be up there feeding. The reason they don't stay up there is because they're also very vulnerable to being eaten by predators. So when it starts to get bright again, they move back down into deep waters, effectively to hide from their predators.
This is where the light-producing photophores embedded in their skin, the lanterns that give them their name, come into play.
These specimens are in ethanol, so they look a bit different. If they were out of ethanol and fresh, they would be kind of reflective. They would look like little mirrors, and they're these little dots that you see on the underside of the fish, effectively on the fish's belly. There are some ones also on the side of the body of the fish. Most of them are on the lower portion of the fish, the part of the fish that when it's sitting in the water is pointing down in the water. We think that those ones, particularly on the underside of the fish, their primary purpose is probably to help conceal the animal when there's a little bit of light. If you can imagine a fish sitting in the water and a predator coming up from underneath it, that predator will see a shadow or a silhouette of that fish against a brighter background. If that animal can produce light from its belly that matches the light coming down from the surface, it can effectively hide in the background. So that's one of the things these animals are probably doing and why they have these photophores.
Okay, so it's all a careful dance with different amounts of light, whether they're moving up and down, and then also matching what's on their belly to the light that's coming through.
That's right, yeah. So it's precisely why these animals move up during the night when it's dark, and why they don't go as far when there's a moon out. So when there's any kind of brightness coming out, they seem to be very good at detecting the light coming down from above and matching the output of their photophores to perfectly match that. But there's a limit to how much light they can produce. So if they're getting into an environment that's brighter than the light they can make, they stop. And they won't go any further.
That's incredible.
It is incredible. And some interesting studies that have looked at this even during the daytime, where these things will move to a particular depth. And if a cloud passes over the sun, these animals will move up and match and basically stay in the same light environment. And when the cloud passes over the sun, they move back down to the depth. So they're very responsive to the light environment that they're in.
It's the lanternfish's night movements, responsive to moonlight, that Jeff is particularly interested in, because they're known to snack on reef fish larvae. In a typical reef fish life cycle, the eggs hatch into a larval stage that lives out in the ocean, becoming part of the mixture of critters that many other organisms depend on for food. When they grow to the right size, they make the return journey to the safety of the reef to mature into juveniles and then adult fish. And they normally do this journey at nighttime.
I'm a reef fish ecologist, and I study these early life stages of reef fish, in part because it's I would argue, the most important stage of the life cycle, where small changes in survival rates can have big effects in terms of populations. So if we can understand kind of what's driving variation in the survival of baby fish while they're out at sea, that translates into big effects down the road in terms of the size of a fish population or the biomass of a fishery that we can harvest. So a big focus of fisheries ecology is to try and understand and predict good times and bad times for larval fish, because that translates to maybe 5 or 7 years later, good or bad years for fisheries.
And he thinks the lanternfish might just be the answer to a puzzle he's been looking into.
One of the things we started to notice were these kind of weird patterns in growth rates of larval fish that seemed to follow a lunar cycle. So we would get this very strong lunar growth pattern. And in trying to think about what would be driving that, our first thought was, well, maybe these fish themselves are able to see at night and feed, and so maybe that's part of the story. But that the timing wasn't quite right to explain that. And in kind of doing the thought experiment a bit deeper, the thing that rose to the surface were these lanternfish that might be driving this. So that's kind of what got us into lanternfish. And then we started looking at lanternfish and saying, these are fascinating animals. We don't know anything about them, or I personally don't know anything about them. And not a lot of fish ecology or physiology has been done on lanternfish. So it's mostly, these animals have mostly been studied by oceanographers who use different approaches. They're working far from shore. They're looking at echo sounders and just estimating kind of biomass and noting that they move up and down. Very few people were actually kind of thinking about what the implications of that up and down movement might be, aside from carbon movement, but certainly not connecting them up to the animals that I've historically studied.
Jeff's mentioned carbon there, and we'll get back to that in a bit. But first, how do you go about investigating this potential link between lanternfish movement and reef fish larvae? While we're catching up in a small meeting room in Wellington, 2 postgraduate students from Jeff's lab, Alisha Gill and Niamh Smith, are doing fieldwork in Moorea, an island just off Tahiti in French Polynesia.
All right, we've loaded up the boat. What are we forgetting today, Niamh?
I think we aren't forgetting anything today, which is surprising, so we're probably forgetting what we forgot.
Yeah, we'll find out soon, right? We've got our cooler for our samples. We've got our mask, fin, and snorkels. We've got our data sheet. We've got all of our safety gear.
We have keys and fuel.
All right, well, should we get going?
Let's get going.
They're staying at a University of California research station on the island. Jeff was there with them initially to get them set up, and now they're running the research themselves, and have sent back a few voice notes of what their days are like. They've got different focuses for their PhD research, and it's Alisha who's investigating those predator-prey interactions between the lanternfish and the baby reef fish.
So we're on site now at our second net, and it looks pretty good. Niamh is filling up our cooler with some fresh seawater, and we will hop in to unhook the net from the reef crest here and bring it back on board.
Like many other coral-ringed islands, there's a flat reef or lagoon area close in towards the island, and then a reef crest, a high point of the reef a bit further out. This is like a ridgeline surrounding the island before the reef drops off into deep water. It's essentially the entrance. To return to the relative safety of the lagoon to mature, the baby reef fish need to wash over this crest. The net she's collecting was deployed the day before to capture what came in that night. Later, Alisha sorts through and identifies who has shown up.
We just got our net back to the research station and have emptied out our cod end into the water table here to see what we caught. So usually there's quite a bit of algae. Today we have a little bit more than usual to sort through. A lot of it is sargassum. And so our first step is to kind of sort through all of that so we can find our fish. So to make it a little easier on ourselves, we start by taking the larger pieces of algae and kind of agitating them in the water a bit to shake anything loose, and then we just double check it to make sure there's no fish caught in there before we then put the algae into a bucket. It's important for us to get a measure of how much algae we're getting each night so that we can kind of tease apart what factors influence the fish we see in our nets. So things like moon phase, light levels, wind, waves, water flow, and amount of algae are a few of the things that we're keeping track of just to see what might impact the fish we get.
On the sites where they're using the nets, they've also used a fish finder, a sonar device that sends out sound pings that reflect off fish in the water. But instead of pointing it down from the bottom of a boat, this one they've anchored to the sea floor pointing upwards to get a general idea of predator movement into the area each night. Alongside this, they have a very sensitive light level monitor, tuned to moonlight. And Alisha is investigating across 2 full lunar cycles.
So once we get all of the big algae out of the way, we can start picking through what's left and sort through the fish. So right now we are right in between new moon and full moon. So we're expecting to get a decent amount of fish, not as much as we would get around the new moon. We usually get a pretty cool diversity around this time. We see things like surgeonfish, squirrelfish, a lot of cardinalfish usually, some puffers, and even lanternfish occasionally. So we pull anything that's larger than about a centimeter out of the sample and keep all of the living fish in an aquarium so that we can do some experiments on their behaviors later in the evening.
The fun never stops on fieldwork. Her job for the evening is to test how the living fish react to a kind of perfume she's developed, essence of lanternfish. She wants to test if the scent of the lanternfish in the water is something that drives these larvae in towards the reef. And in case you thought doing fieldwork in Morea is all totally dreamy, that's very heavy rain that you can hear in the background.
Now that we've got everything that's over a centimeter out, we're left with a bunch of smaller bits of algae and really tiny, tiny fish. So these tiny fish, there's so many of them, and it would take hours for us to count every single one that we have in this sample. So what we do is we take a subsample of the table so that we can make an estimate of how many there are. Some nights there's over 4,000 tiny fish, so you can imagine why we do the subsample.
Why Morea? Well, Jeff has been researching in this region for a long time, so he's got a good understanding of the timing of when the fish larvae return to the reef. On top of that, the very clear water means more moonlight can get through, so they should have a good strong light signal to pick up to help address their hypothesis. In particular, he's interested in the nights where there is a moon, so it should be bright, but then some cloud comes in.
And in those times, we would expect there to be kind of an anomalous increase in the numbers of kind of predatory fish like lanternfish that might be coming into these shallow waters. And we would also expect to see kind of an anomalous kind of rush of these larval fish basically fleeing that predator group, kind of coming in and returning back to the reef, perhaps younger than they might otherwise return. So that's some of the sort of things we're looking at. We're looking across a lot of different species, and we expect those sorts of patterns to be particularly prevalent in species of reef fish that are small and relatively undefended. We are also getting a bunch of species in our nets that are larger and have spines and are basically defended from predators. They have armor. And so we have an expectation that we should see different responses. If you're vulnerable to lanternfish, you should be exhibiting these responses and responding to this landscape of fear and returning to the reef. If you're immune to these lanternfish because you've got big spines and they can't fit you in their mouth, then you probably don't care and you just do whatever you're going to do.
They've also collected lanternfish samples from across the Pacific by teaming up with other expeditions. More locally, they've partnered with Ngāti Pahuera in northern Hawke's Bay to explore whether the lanternfish movement is part of Māori traditional knowledge around the lunar calendar.
Here in New Zealand, we have Maramataka, which is a very important aspect of Mataranga that shapes activities around the lunar cycle, and particularly those activities as they relate to fishing could well be influenced by the movements of these lanternfish, for example. Even if lanternfish weren't a historic resource for Maori, the effects they might have on coastal species might be sort of incorporated into this Maramataka. So that's another aspect of the work that we're very excited about exploring.
In case you're wondering, people don't tend to eat lanternfish because they're small and bony and super oily. Just really unpleasant. And they can cause digestion problems. But there are some commercial fisheries around the world harvesting lanternfish for use in fish meal and animal feed. This has sparked debate because the other role that lanternfish are thought to have is a helpful one for us, moving carbon from the surface to the depths of the ocean. And because there are so many of them, it could be a big role. The carbon cycle starts at the surface, says Jeff, where carbon moves from the atmosphere into the ocean.
A lot of that carbon gets taken up into the surface waters of the ocean, and it's also sort of incorporated into microalgal material. So
that's photosynthesis.
So photosynthesis is locking up carbon in animals, and it enters the food chain that way. So things eat the carbon, and bigger things eat the phytoplankton, and lanternfish are one of those. And so by moving up to the surface and feeding on it at the surface, and then moving back down to deeper water, and potentially releasing that carbon via respiration or via making fecal pellets or being eaten by something at depth, they're transferring carbon faster than it would otherwise just sink by passive processes.
Maybe they poop it out, or swim it down, or decaying remains of animals drift downwards. A lot of the sinking carbon is recycled back to the surface 100 to 1,000 years later by the ocean circulation. But if it reaches the seafloor and gets buried in sediments, it can be sequestered or locked away for a very long time. Eventually, over millions of years, it could be incorporated into rocks or turn into gas or oil. And some of the lanternfish might go to depths of 1,000 meters, says Jeff, which might be a great help in locking carbon away.
The time frame, I think, of carbon just sinking passively is probably hundreds to thousands of years. So it's a long time to just passively sink, but these animals that are moving up and down can short-circuit that and make it happen much faster.
And so is this a kind of idea or hypothesis at the moment, or do we know that this is happening, that they are moving this carbon down into the depths?
We know that they are consuming carbon and they're moving up and down, and there are some assumptions about what the fates of those animals are that go into models of projecting future climate, for example. In those models, this pathway of active movement of animals is probably the one that has some of the most uncertainty about it. Actually, we have estimates and those go into the models, but we're not sure about some of the details. And so that's where our work is kind of coming in, trying to understand some of those details a bit better.
It's kind of crazy to me that the daily commute of these small shiny fish are being worked into our climate models. But again, there are just so many of them.
If they move up and they feed and they move down and they poop, they release their carbon at depth, then that constitutes a really big pathway for carbon to be moved actively by these animals. And that's an assumption that happens. We have reason to suspect, or we hypothesize, that these animals may not be doing all the behaviors we assume they're doing. And one of the reasons is that when they undergo these migrations, they move between very cold waters at depth and warmer waters at the surface. And we know from a lot of other animals that their metabolism, the rates of metabolism, their digestion, things shut down when they enter cold water. So one possibility is these things might be moving up and down, but not moving carbon, not releasing carbon at depth in the same way that we assume.
If they get eaten at depth by something that lives down there, well, that's bad news for the lanternfish, but good news carbon movement-wise. But as to whether they poop at the surface or at depth, it's a tricky question to answer. Your best bet might be to collect some lanternfish and chill them down to simulate that cold deep water, and then check what happens to their digestion in those conditions. But
one of the challenges with studying this species, and one of the reasons we don't know a lot about it, is they're very difficult to keep alive in captivity. And a number of reasons for that. One of probably the most common one is that we tend to catch them in nets, and they tend to come up out of these nets quite damaged, and they seem to be quite fragile. So any little bit of damage seems to make it so these animals are not likely to survive very long in a tank or a bucket or whatever you put them in. So we're trying to change the way that we catch them, to catch them more gently, to kind of remove that effect. It may be that we can't estimate those things directly by observing live animals, so we're trying to develop a kind of a forensic approach to trying to reconstruct what they might be doing in these different environments.
this forensic approach involves doing some chemical analysis of the fish's ear bones, or otoliths.
That's one of the really cool things about fish, is that they have these structures in their inner ear that, for the fish's benefit, they help with hearing and balance, but for our benefit, they record things that are in the water chemistry on a daily cycle. So we can extract these ear bones from a specimen. We can prepare the sample in such a way that we can try to measure the chemical profile across a series of days to try and get a sense for where the animal was, what it might have been doing, potentially its respiration rate or metabolic rate over those days.
In Morea, it's Niamh who's focused on this carbon movement work. Their next field trip will be in quite a different setting, in October across a new moon off the coast of Otago, with their collaborator there, Dr. Steve Wing. And part of the reason for sampling across the Pacific and around New Zealand is to investigate whether lanternfish metabolism and digestion rates differ in different ocean temperature ranges. Overall, they're hoping their work will lead to better understanding of how the rhythmic dance of these small fishes shapes our oceans, from the fish we eat to the Earth's carbon cycle. Thanks to Professor Jeff Shima, Alisha Gill and Niamh Smith of Te Heranawaka, Victoria University of Wellington. This episode was produced by me, Claire Kincannon, with production help from Ellen Rikers and sound engineering by William Saunders. You can find more information and pictures on our webpage at rnz.co.nz/OCW. And if you have any feedback or questions, you can email us, ourchangingworld@rnz.co.nz. If you don't already follow the Our Changing World podcast, then I recommend you do. It's the best way to make sure you never miss an episode. And if you like deep diving into ocean mysteries, I can highly recommend the recent RNZ original podcast called Kelly Tarleton's Final Treasure Hunt. Find it wherever you find your podcasts. Tēnā koe I whakarongo mai. Thanks so much for listening. I'm Claire Kincannon. Have a great week. Kia pai, te wiki.